Thank you, and welcome to the webinar discussion on Epicatechin, which is targeting the mitochondria as a potential therapy for muscular dystrophy, and specifically in our case for Duchenne. Thanks to all of you for joining. We're joined today by George Schreiner. I won't read his extensive biography, but I will say to you that he has received his MD PhD from Harvard. He's then served for a long time as an instructor in pathology and assistant professor at both Harvard and Wash U St. Louis, and then went into industry. So he is really well versed in both medicine and industry, and he's here to talk today about Epicatechin. For all of you out there, there's lots of questions that we have around Epicatechin, and we're going to ask George questions along the way. And more specifically, George, as this community is well aware, there was a study on negative Epicatechin that's posted on clinicaltrials.org. So in the course of your conversation, we'd like you to address the differences. So with that, I'll ask you to begin, George.
Good afternoon, and I want to express my appreciation to Pat and the Parent Project for giving us this forum as an opportunity to answer your questions. We're in an odd position, which is that while we have some very exciting data, we're a very small new company, and we are advancing a medicine that is completely novel with respect to its mechanism of action. And so there's not very much known about the role of Epicatechin in muscular dystrophy, except for the work that we've recently finished with Dr. McDonald at UC Davis, and we'll be continuing with him and other investigators with the support and collaboration of PPMD. So I just wanted to run you through this, and this is coming at your children from a novel angle, and I think it's a very important piece of new biology. I will emphasize that we did not start off attempting to make a drug for muscular dystrophy. We were actually looking at cardiovascular disease. I'm not a pediatrician. I'm a nephrologist, but we were so astounded by the biology of what we were seeing. And we were seeing such striking effects on muscle as well as mitochondria that the science actually led us to muscular dystrophy. And once we listened to what the data was showing us and understood what we had uncovered, muscular dystrophy became far and away the most obvious target where we could have the greatest impact in the shortest period of time. So we are very, very excited about working in this space. Like Blanche Dubois, we are very dependent on the kindness of strangers, and among the most helpful to us has been the PPMD in bringing us up to speed on the unique aspects of developing drugs for orphan diseases like muscular dystrophy.
George, sorry to interrupt you, but please speak close to the microphone so we can record.
Okay. All right. So I and my partner have a lot of experience in drug development, but not in this space. So this has been a true partnership that we are establishing with PPMD in order to get these compounds into the hands of the physicians and your patients. So starting off with the slides with gene mutations, Daniel, that's the second slide. I'm not going to review, because you all know this very well, that numerous gene mutations have been defined as contributing to the abnormal functioning of muscle sarcoglycans. These are the proteins that sit on the membrane of the muscle cell and connect to the extracellular space, the bones, and to the tendons that allow one to move one's muscle. And when this is shown in the next slide, slide 3, I'm sure you've seen this many times, and this is a cartoon of the sarcoglycan complex, and mutational errors in any one of those can cause muscular dystrophy of varying severity and varying expression of symptoms, but they have this universal underlying mechanism. What hasn't been appreciated in muscular dystrophy is that this abnormal structure of the muscle with which muscular dystrophy patients wrestle every day of their lives has downstream effects on other organelles or other parts of those muscle cells that we think play an equally important role in the idea of progression. This is an important concept that's sometimes not emphasized enough. Muscular dystrophy, at least in the most broadly expressed form of Duchenne, has a period of time of relative compensation and then a relatively abrupt deterioration and progression of weakness, and often occurring between the ages of 6 and 10. Now, what's happened in that period of time when these kids go from being able to walk to being wheelchair bound? The mutation hasn't changed. They haven't put on the muscles of puberty so that they're overloading their frame. But what's happened is actually a discovery that is as old as the discovery that dystrophin is the primary protein, or is the protein mutated in Duchenne's, and that is that as these patients start to deteriorate, they start to lose evidence of mitochondria presence and function in their muscle cells. So let's talk about mitochondria as an important component in why this disease gets worse, not only progressing to paralysis, but also to the failure of the most dynamic muscle of the body, which is the heart. So on slide 5, there's a slide that says the anatomy of a cell's power. So you see this familiar depiction of a cell with the nucleus, and it has chromosomes in that nucleus, and on those chromosomes can sit the mutation for Duchenne and other muscular dystrophies. You see the endoplasmic reticulum where proteins are made, and the Golgi apparatus where they're folded for release into the circulation or for placement in the membrane. And you see those little orange pill-shaped things called mitochondrion. That's the singular. Mostly it's people refer to mitochondria in the plural. Now, as is summarized on the next slide, mitochondria are the source of more than 95% of the ATP, which fuels every activity in the cell's compartment. Nothing happens without ATP. There's no contractions. There's no functioning of the nucleus. There's no transport of ions in and out of safe places. The whole system comes to a crashing halt in the setting of ATP depletion. And mitochondria, like any furnace or power plant, they wear out on a regular basis. They have to be replaced roughly every 2 weeks in our body. But if there's an ongoing process, such as muscular dystrophy, they decay faster. And although there's a lot of mitochondria in the average muscle cell, 10,000, not just 1 or 2 as you see in these cartoons, but there can be 10 to 15,000 mitochondria, with the highest density being in skeletal muscle, in heart muscle, and in brain tissue, okay, or neuronal tissue. And if there's an additional stress, the mitochondria cannot be replaced efficiently. And this replacement requires this fancy word called mitochondrial biogenesis. Genesis means the creation of new biologically active mitochondria. It's a term that I'll use again. There's not a single drug that induces mitochondrial biogenesis before our compound that we're advancing into the clinic. And when you lose your mitochondria, when you have inefficient or blocked mitochondrial biogenesis, then that results in the weakness of the tissue, and then eventually it's atrophy or conversion into scar tissue. And as you might guess, loss of mitochondria is associated with many diseases of aging. But in this case, we're talking about damaged mitochondria and loss of mitochondria in children. And how do we get to that point? On the next slide, where you see it says unhealthy mitochondria accelerate tissue damage, Healthy mitochondria, depicted on the left, make lots of energy. That's ATP. Now, in the process of making ATP, some harmful oxygen radicals are made, but not many. And the mitochondria, a healthy mitochondria, has the capacity to neutralize those oxygen radicals so they don't destroy the tissue surrounding those mitochondria. In an unhealthy mitochondria, and an unhealthy mitochondria means unhealthy cells, the mitochondria are relatively less efficient at producing energy for the muscle fibers to use, but they produce lots of harmful free radicals, and those radicals go on to destroy the muscle fibers that are lying very, very close to the mitochondria and the ATP, because the way the system works is the mitochondria right up against those muscle fibers, they release ATP, the ATP goes right into the actin and myosin of the muscle, they contract, you lift your arm or you take a step. And if those damaged mitochondria are now making a lot of oxygen radicals, they're not only going to damage themselves, they're also going to damage the muscle tissue that's lying very close to it. And you can see in the next slide, mitochondria surround and support muscle fibers. The mitochondria are those things that look like fingerprints, and the muscle fibers are the stripes. And, you know, you're looking at, you know, the essence of a heart muscle. There's not much room for things other than mitochondria and muscle fibers, okay? And you can't have one without the other. And if you lose your mitochondria, you start to lose your muscle fibers because they're not working, and they tend to be eaten by the cell if they're not working and broken down. And the net result is what we see in our older kids, which is the inability to function in our with respect to their muscles, the requirements for wheelchair, and the progression towards heart failure. So everything that you know about, or virtually everything you know about, with some exceptions, but 90% of the drugs that are being tested in muscular dystrophy are trying to target the defects in those stripes of muscle fibers that you see on that slide. We have the only approach to trying to improve and restore those big mitochondria that are essential to keep those muscles going. We're not going to cure muscular dystrophy, okay, but what we think we can do, if what our early data is positive, is that we may be able to freeze it or reverse it back to the earlier stage when patients could use their arms and their legs and were not necessarily progressing to heart failure. And we are even more excited about the possibility, which we haven't gotten to yet, that we could combine this oral intervention with, in fact, gene therapies, because we know that, in fact, this stimulates the expression of the proteins that the gene therapy companies are trying to stimulate, with their nucleotide material. So we see this as a potentially profoundly game-changing way of preventing progression, potentially reversing by restoring mitochondria, and synergizing with other strategies to enhance the expression of utrophin or dystrophin. Now, I told you that there are thousands of mitochondria, and in the next slide, you see this beautiful picture of the way the mitochondria line up along the fibers of muscle cells and tissue culture, and they look just like what they are. Brilliant energy pulsing through these cells that are contracting. They're sticking to the matrix of the cell. They're being active, and these little green bursts of energy are in strings along the cell, and they are what allows a muscle cell to be a muscle. And if you don't have mitochondria, even if you have all of your sarcoglycans, you're going to be severely impaired. What is the first thing that goes when you break an arm and you have that arm put in a cast, or you break a leg? The mitochondria, which are not being used because the leg is immobilized, disappear. They are expelled into the circulation when they're not being used and the muscle's not being contracted. And that's why when the cast comes off 4 to 8 weeks later, those muscles are shrunk and weakened because they've lost their mitochondria. So they've got everything else they need, but without those mitochondria, they're not functioning. And the whole process of rehabilitation after that kind of trauma is to build the mitochondria up. And to this date, the only known mechanism to try and enhance mitochondrial density was exercise. The problem with exercise is that it generates excessive oxygen radicals. So it can be damaging if you have an already damaged muscle and you do too much exercise or for too long. But what we believe that we're working on is the hormone of exercise. So you can get to the point of building the muscle up in a physiological way, but by taking the hormone that's released, and we'll get to that in a minute. So why is mitochondrial density critical to the progression of muscular dystrophy? So this is on the next slide, and it says that there are a variety of muscle glycans, sarcoglycans that are damaged by genetic abnormalities, but they all share a common pathway of muscle atrophy, and that is that there's metabolic damage to the mitochondria and a depletion or loss of mitochondria over time. And this correlates very nicely with the progression of weakness and the development of heart failure and muscle atrophy. And in fact, the understanding of this is as old as the discovery that dystrophin is the culprit in Duchenne muscular dystrophy. And in fact, Brian Hoffman's lab was one of the first to show that there is a loss of the energy fuel generating source in the cell together with the loss of the of dystrophin. They go hand in hand. They are linked functionally to each other. The mitochondrial injury is lost to the excessive contractility. As these kids are trying to move their muscles, they're working their muscles very hard, much harder than normal kids have to, healthy kids have to move their muscles. And this results in the accumulation of calcium, which is toxic to mitochondria, and a lot of oxygen radical accumulation from mitochondria that are working overtime. And so there's a progressive loss of mitochondria. The problem then being is that the smaller number of mitochondria are having to work even harder. And the harder they work, the hotter they burn. And the hotter they burn, the faster they die. And so the net result is that a 10 or 15-year-old with muscular dystrophy has a lot fewer mitochondria than when they were 5 or 6. And the kids' normal mechanisms for replacing mitochondria, mechanisms that should be peaking at their age and don't start to decline in healthy humans until the mid-20s, are not enough to compensate for this extra trauma. And so they lose not only the muscle energetics, but they lose the capacity to repair their muscle, which we have found closely linked to the mitochondrial presence. And here is why we think that Epicatechin on the right may potentially improve function and slow the progression of myopathy or the muscle disease that is associated with the gene defect of muscular dystrophy. All right, next slide. What's the science? Well, Cardero, and what's the warning, by the way? I need you all to pay very close attention to this. I know the way everybody is feeling on this call because I have a daughter with a disease that every time discussion of a cure or a therapy comes up, she got it when she was 3. I leap to the internet and I start making phone calls. So I'm going to advise you not to overreact to what I'm about to tell you, because there's no shortcut in this process. We found that this molecule, the epicatechins, and there are 2 main ones, they're members of a family of natural products called flavonoids. And these are actually found in cocoa pods and in tea leaves and in some other plants. And we showed some years ago that these are the most powerful stimulators of new mitochondrial formation in disease states when we give it isolated and purified. All of those compounds that I just showed you have over 500 to 700 molecules in them, okay? Many of those molecules inhibit the process of epicatechin stimulation of mitochondrial proliferation. So you cannot put your children on lots of chocolate or lots of tea and have any expectations of a therapeutic benefit. So I do not want you to run out and do this. What you need is purified material. We have the world's only pharmaceutical-grade synthetic material approved by the FDA already in our earlier trials. It's much, much more active than anything that we can extract in any way from plants, even when we try to purify it. And this is the way to go in order to optimize the response of kids with muscular dystrophy. Okay. Now, why does this work? Well, there are a number of medicines that have come out of plants that work because they are mimics of molecules that are already in your biology. The classic example of this is the opioids. Morphine comes out of the poppy seed. Morphine and its derivatives, which relieve pain, work by acting as analogs to endorphins. They act upon the endorphin receptor. And the endorphin receptor, which mediates pain relief, is an endogenous system that we all have that nature provides us to get through severe chronic pain. And it's a biological accident that in the 600 billion botanical plant products that are known, a set of them from a particular poppy that tends to grow in Southeast Asia makes a molecule that mimics endorphin, binds to these receptors, and relieves pain. Digitalis, which for heart failure is a plant product out of foxglove that resembles molecules released by the heart to facilitate or improve heart function. There are many, many examples of this. Taxol, the drug that came initially from bark and turns out to be very effective in breast cancer cells. So this is the next in that line. These are a natural product that is just been found to mimic a hormone that Cardero has also discovered. It's the first new human steroid hormone discovered in half a century, in which we have found that it is released in exercising muscles and put into the circulation, where we think it's mediating the beneficial effects of exercise in other tissues and in the brain when you exercise. But the most important feature for you to know, and we're going to be publishing this in the next 6 months, is that it is released by mitochondria. So it's what's called an autocrine hormone. The mitochondria, when they're physiologically stressed, not in disease, but in an exercise, release this hormone to make more of themselves, okay? What's the problem with a kid with muscular dystrophy? He's losing his mitochondria. He can't make this hormone, not because there's anything wrong with the mitochondria. He doesn't have enough of these mitochondria to keep his hormone levels at a normal level and thus to keep his mitochondrial density up. So it is our belief that we can provide this and basically allow a 6 or 7 year old or a 14-year-old or a 20-year-old with muscular dystrophy to have the muscle hormone driving new mitochondrial formation that a healthy kid has, and that these kids are basically lacking, okay? And that we think as we stimulate mitochondria and they start providing more of this hormone on themselves, that in fact we'll be able, we hope, to bring the whole muscular system back into a healthy equilibrium. And that's obviously just a dream right now, but we've got a lot of suggestive evidence that this is possible. Now, we've been discovering a rare form of epicatechin called the plus form, which is so rare that it's not available for sale, even for research purposes. We have made the world's only supply, and it's 400 times more potent than the form of epicatechin that one finds in tea or cocoa. So as we move from the Becker's muscular dystrophy, which is a less severe form of muscular dystrophy, to the more severe example of Duchenne's, we are moving to this extremely powerful form of epicatechin for which we have the world supply right now. And you cannot get this, by anything that you can go online and look at and buy. And we think it's going to need something with this kind of punch in order to make a difference in the severity of the illness of the kids with Duchenne. And we think that because with Becker's we saw a big impact on the heart failure of the older Becker patients, that this more potent version has a lot of promise to us for dealing with the heart failure of older Duchenne's patients, which is not something that is for which we have a lot of therapeutic tools right now. Now, if you want to see an example, and this is what we realized that we had to focus on muscle disorders, we looked in the next slide where it says Epicatechin restores leg muscle structure of elderly diabetics. We looked at severe diabetics who basically led a bed-to-chair existence. We gave them Epicatechin for 12 weeks orally, and on the left-hand side you see their muscle biopsy before, and on the right-hand side you see it after. The little M's you see are mitochondria. The muscle on the left looks like spaghetti. It's got no particular disorder, and the mitochondria look like these little fragmented, swollen bits of tissue. There are virtually no normal mitochondria in that field under that microscope. If you look on the right-hand side, you see two remarkable things. You see all those little gray bean-shaped things. Those are all the mitochondria, which have now been restored almost to normal in these 75-year-old men who could not walk more than 10 yards, okay? And this particular gentleman was walking 1 mile to 2 miles at the end of this therapy. But just as importantly, we are also seeing a restoration of the normal organization of the muscle. And these men were making more muscle fibers, and they were making more sarcoglycans. In fact, they were making more dystrophin and utrophin. So they were regenerating their muscle at the same time that they were acquiring the ability to use that muscle. And we looked at this and said, holy cow. We said, we've got to focus now on disorders where this is not a 75-year-old man with diabetes that we're trying to help. It's an 8 or 15-year-old boy who is watching his muscle structure slide out from under him. So we've published a number of papers. Haven't published our human data yet because this is breaking news essentially. But you can see on the next slide the summary of the effects of Epicatechin on muscle, specifically in our rodent examples and also in our more limited human examples. We've clearly seen in numerous experiments increased mitochondria in both skeletal muscle of humans and rodents. We've seen it in leukocytes, showing that it's universal to other tissues. And in the animal models, we've seen it in heart, brain, and liver. And the heart response is extremely prominent. We've seen evidence of muscle regeneration and improved muscle strength in both humans and animals. And very, very interesting to us in what is currently a very hot area right now in muscular dystrophy, in which people are trying to decrease a muscle atrophy hormone called myostatin by using antibodies and other means, or they're trying to stimulate a muscle trophic hormone, a muscle stimulatory hormone called follistatin. Our compound, which is taken by mouth, actually both stimulates follistatin and it lowers myostatin. So part of what we're seeing in the muscle and part of why we're showing a coordinated response between the contracting parts of the muscle and the energy production parts of the muscle is that we're doing just that, but we're doing this in the context of replacing the energy loss in addition to replacing the structures that are being regulated by follistatin and myostatin. So we're very excited, and that we know that to the extent that any of the trials that are being done show that decreasing myostatin or providing follistatin help, we know that Epicatechin will help because it has that as part of its therapeutic effects. All right, so we have some data in muscular dystrophy, Becker's muscular dystrophy, which is more of a quantitative defect in dystrophin expression, less qualitative, and these patients develop many of the abnormalities of Becker's, but at an older age, but they also become progressively weaker and have a high incidence of heart failure. We did this with Dr. McDonald at UC Davis. And on the next slide where it says functional responses in Becker's muscular dystrophy after 8 weeks, and in this we were using the weakest form of epicatechin, the minus. If the patient showed some muscle, that is, if they were not end-stage muscle atrophy, they all showed improved 6-minute walk test. This is 8 weeks. 3 of the men had functional responses that were meaningful in their daily life, including throwing away a cane that they had to use on 5 years. All of them showed improved exercise tolerance. All of the patients, even the ones that didn't have an immediate mitochondrial response because their muscles were so badly damaged that we realized we'd have to treat them longer, we think, to see a better response. All of these patients showed improved blood oxygen content in the blood because epicatechin dilates blood vessels. They all showed lower lactate levels, which is a sign of improved mitochondrial function. They all showed improved oxygen efficiency. So that meant they all tolerated exercise better at high levels of exercise after only 8 weeks. They all showed improved heart function during exercise, and they had a 75% reduction in a marker of BNP, which is a marker of heart failure. And you can see in the next slide that all of these patients, the normal level of BNP is in the red. All of these individuals had more or less degrees of severity of heart failure. And in 8 weeks, all of those markers of heart failure went down as we saw evidence of improved cardiac function. To my knowledge, and I may stand corrected on this, but this is the first demonstration of reversal of established heart failure in anybody with a muscular dystrophy. Doctors have shown that if you start a youngster early on heart failure drugs at the age of 10 or 11, you can delay by a year or two the onset of heart failure, but virtually all children have some degree of heart failure by the time they're 18. I don't know of anything approaching this kind of response in patients that already have pretty advanced heart failure. So we're almost at the end here. So what are our plans? Well, we're going to take this 400 times more potent version of epicatechin, and we're going to be looking now at a group of patients that are not often addressed in clinical trials. And I have to say that this is part of the leadership of PPMD. They really highlighted for us, although we certainly intend to have this drug being developed in parallel for children as a preventative. PPMD was really critical for us in saying, we need something for the 10 to 20-year-olds, 10 to 25-year-olds. Santhera is doing something with that age group in terms of respiratory function. But this is an unserved population. And we already knew in our older Becker's patients that they seem to be quite responsive to this intervention. So we're going to be targeting muscle function and heart failure. We're going to be looking for improved capacity for exercise. We're going to be looking for relief of nocturnal dyspnea, the breathlessness that Duchenne patients often experience when they're lying down. We're going to try and maintain or perhaps increase the range of motion of arms to maintain the quality of life of the older patients with Duchenne. And we're going to see if we can reproduce our little pilot experiment, but now with larger numbers, to see if we can reverse the severity of heart failure. So we're looking at everything from improved quality of life to, in fact, even, I believe, the potential for prolonging life, because I think this is ultimately what these kids die from. The trials that are being planned is, and the first one is being supported by Parent Project, is a dose-ranging safety study of plus epicatechin. We're going to be doing this in the earlier stages of non-ambulatory DMD. They have early stage but not clinically significant heart failure, and they have moderate loss of arm function. We're going to be doing this for 8 weeks, a lot of biomarkers and direct tests for strength and energy and exercise. And then we're going to extend that study into a phase 2 study that's going to enable us to then go for a phase 3 trial. The other thing that we are sharing with PPMD is a shared commitment to getting this into the FDA approval process as fast as we can, because people don't have the luxury of waiting for us to take a long time to get this proven while their own kids are getting sick in front of their eyes or progressing in their illness. So we're now going to take the sicker kids in the later stage non-ambulatory Duchenne, now with clinical signs of heart failure, clinical signs of respiratory insufficiency, and we're going to look at, if you see the first trial, it's focusing on prevention. We're now going to focus on reversal and slowing the progression, reversal of heart disease and slowing the progression of the muscle loss in these non-ambulatory patients. And then, depending upon which population responds the most to us, to the Epicatechin, we're going to focus on that for drug approval, but we're going to be bringing along the other groups right behind it and expanding that. If we confirm the scientific hypothesis that we're testing right now, that this new class of therapeutics is going to add a heretofore unexpected arrow to the quiver of doctors taking care of patients with muscular dystrophy, then there's absolutely no reason, as you can see in the next slide, why this potentially, and remember we're still talking about a dream here, why potentially this couldn't address the entire spectrum of Duchenne's independent of the gene defect, because loss of mitochondria causing muscle atrophy is a universal mechanism. It happens after orthopedic surgery. It happens after hip replacement. It happens in other diseases, not Duchenne's like ALS or Friedreich's ataxia, where loss of mitochondria results in paralysis of muscles and loss of use of muscles. So we are addressing a universal mechanism of muscle failure, and we think, therefore, it'll be applicable no matter what the gene defect is and no matter what the age. We've just got to figure out in the next few years best doses, best duration of therapy, and if there are any safety issues, how do we prevent them or look for them? We think that this is potentially a standalone therapy. It's very safe. There's a very safe class of molecules. It's in the diet and for many people. But we also think it's highly complementary to the current Star Wars era of Duchenne's muscular dystrophy therapy. So exon skipping approaches, if we're boosting dystrophin expression in cells that have that capability, and now you're giving cells that capability through exon skipping, then we think it's a reasonable hypothesis to test. Can we take a moderate effect and make it a much bigger effect if we give both? So that's something that we've definitely got on the table as we get through these early critical studies. And we have the only intervention that seen in both animal models and in our limited human pilot data that seems to link improved exercise capability, provides evidence of muscle regeneration, and improves cardiac function. That triad, which is the triad of defects in Duchenne's, is being addressed by a single compound. And that's not to say that we won't need more, but it's a unique feature of this novel class of therapeutics that are targeting not just the mitochondria, but the mitochondria appear tightly linked to also maintenance of muscle structural health. So in the last slide, speed into the clinic and into the trials, getting FDA approval is the number one priority of our little company. And because we're dealing with a pathway that's never been discovered before, we need clinical data to bring in the larger scale of financial support that's going to allow us to drive this towards registration trials and drug approval trials so that we can get this into the marketplace for patients and their physicians. Here is where PPMD is playing the role of guardian angel. They're helping us raise money for this critical pilot trial in heart disease and muscle weakness. They're guiding us with endpoints. They're providing all the expertise, not just of their physicians and scholars, but also of their patient families. So for me, who's not grown up taking care of Duchenne's patients, Pat and her colleagues are introducing me to the community and saying, as you're doing this drug development, listen to everybody, because this is what needs to be done. This is what we're looking for. Build it into your trials from the get-go, and together we can make this an effective partnership. So we're starting off on an exciting journey. Science, so I can only hope that what we see next is validates what we've seen in our animal models and our early human trials. We've had minus epicatechin, the weaker form, in over 80 patients. We've seen a lot of things that should benefit Duchenne's, so it's not, we have a lot of experience, and I think this is going to be just a great adventure. And with that, I'm finished.
Thank you, George. That was very, very helpful. We so appreciate it. George, there have been a number of questions that come in, and some of them are around the clinical trial that we're planning. And there was one question about 5 to 9-year-olds who can walk, and as you mentioned, the population is going to be a little bit older and on the verge of loss of ambulation and then non-ambulatory. You mentioned that the trial is 8 weeks long, and we've talked about, and could you see a signal by then? We've talked about using the imaging DMD to take a look at those quadriceps muscles, the big muscles in the upper leg, to see if there is activity. So could you kind of shed some light on the reason for 8 weeks long and what we're hoping to see, and then what the plans are beyond there?
Thank you. Actually, this is a very good question, and it gives me a chance to clarify this. We have our regulatory consultant, Tim Franson, who's worked a lot with your community in terms of developing protocols with the FDA. He actually advised us to do two studies, and one was because we're dealing with sicker patients here, that the first study is 8 weeks because it's a safety and a dose ranging study. So what we're going to be using in the first phase is a range of doses, because we want to make sure that there are no side effects that are novel, that are outside our current expectations. And we're going to be using imaging studies, as Pat showed, of both heart and muscle. We're going to be using biomarkers. We're going to be using direct analyses of muscle strength and cardiac function. But let me emphasize that 8 weeks is too short a time to establish true efficacy. So what we are then going to do is take the optimum dose, and in the second trial that I mentioned, we are now going to do that as a 3 and 6 month trial. And that is really, and the reason for that is that we think 3 to 6 months are an absolute requirement to show regeneration of muscle, and that 8 weeks is just not long enough. So we're going to give the muscle every chance to respond to the epicatechin beneficial signaling. And because we have limited resources in terms of money and amount of drug, because we're making, as I said, we have to make the world supply for every trial, we only want to use one primary dose for that we got from the dose ranging, and we don't have enough resources to do both trials for 12 weeks. But the plan would be to do that sequence, and then for the long trial, the 3 to 6 month trial, use the clinical endpoints that we would reproduce in a phase 3 trial for approval. And so this phase 2 trial would be a stepping stone to an approval trial. The FDA generally does not accept, they accept surrogate markers and imaging for dose ranging, but they tend not to accept those kinds of endpoints in lieu of actual efficacy. So it's a shorter phase and a long phase study. Now, I would love to do 5 to 9-year-olds, and Dr. McDonald is extremely enthusiastic about that. We have two challenges to that. One is that we need to start with a population that can give informed consent, and for the most part, the older Duchenne's patients that we're targeting first can do this, okay? And obviously their parents can do it for the youngsters, but In general, the FDA likes a situation where the people on whom this is being tried first have a better understanding of the risks, if there are any risks, which I don't know yet. Secondly, there is a lot of clinical activity going on in this age group because of the number of trials involving exon skipping, myostatin inhibition, and so forth. We're really a small company that has appeared out of left field. We have more than a half a century of drug experience between the two co-founders, but it's really hard for us to compete with all the big established companies in this population. So even in terms of identifying patients. So we've got to go to them and say, they've got most of the patients that, because this is the age group. So we've got to go to them and say, look at how effective this is in the older age group or the group that doesn't have a gene defect that's being addressed right now. Let us combine it, with your exon skipping and see if we see something much better. And that's probably going to be our best way of getting into that population and eventually being able to study these kids, outside without giving them exon skipping or other kind of genetic manipulation therapy. So it's a harder path for a young company to trot given how many big players there are in that space. Whereas in the group that we're currently trying to serve, we're it. We're the only solution for muscle and heart at this point. So we thought we could get the patients faster, get the trials done sooner, and then start to branch out into the preventative. In general, I can tell you from having been a doctor for many decades, the earlier you start, the better the effect that you see. So we're really committed to moving this upstream as fast as we can so that kids can start on this at the age of 4 or 5. And if it's as safe as we think it looks like so far, this should be very tolerable, very well accommodated by children. So that's the plan, but we had to start somewhere. So we decided to start with an unserved population.
Right. Thanks, George. Yes, we want to start with this. We want to start with this this population that is less likely to be involved in trials based on inclusion criteria. We want to get the dose right. I think that's really important. Make sure that the production of the material of the drug itself or of the compound itself is available. And we've agreed with we've suggested to George, and we've all agreed that the individuals that participate in the study are going to continue in the study as not be withdrawn from the drug for us to start and stop another study. So this is a continuous sort of a continuum that we're going on to advance this as quickly as possible. Some of the questions are around, would this potentially be additive or helpful in terms of combinations of other drugs like utrophin upregulation and should VBP15, the steroid replacement drug of Regeneration, come to have that opportunity? And I think the answer is yes. It could be synergistic with many other compounds to include exon skipping, as George mentioned, utrophin upregulation, certainly the use of steroid replacement or drugs that are impacting the inflammatory response, which is what VBP15 and catabasis drug does. So I think it would be synergistic. Other questions are, have the participants been selected? They have not. We will be recruiting through Duchenne Connect. We are just now working on protocols and trying to really get that set. And as you all know, that once you establish a protocol, then when working with the sites, then we have to go through their IRB and the process to get it approved to start. So I don't have a start time. Let's say we would like to start this yesterday. We will let you know more as we develop the protocols and go through the process at the clinical sites so that you're well aware of the progress, and I promise to come back to you. So first quarter, we're going to be up and running in the study. And then Dr. Steph is on the phone, George, and she asked, because on clinicaltrials.org, or .gov, sorry, there is the Becker study listed by that Craig did with you, and I know you provided some insight into the 10 patients of Becker that were treated, but it says that there is no update. Will there be some formal update that's posted on clinicaltrials.gov anytime soon?
Yes, we're just dealing with a overloaded academic center right now. So it's we don't own the data. The data is owned by UC Davis. This was an investigator-initiated IND. So Cardero provided the drug and the science, but the trial was conducted by Dr. McDonald, and so we can only encourage. We have no access to, no control of the data. It's under Dr. McDonald's control. We have urged him to put the rest of the data on site, and we're working with him to get the first publication on this data published, because it was actually great data. So, but that's not under our control right now.
Okay.
As we get into drug registration trials, then the INDs will be moving over into our control. They'll be sponsored by Cardero, and we will own the data, and we will be extremely vigorous in pushing that data out into the public domain.
Right, and we are working with Cardero as well. We're working directly with Cardero so that we can make sure that this information continues to be available and is available to all of our community. One question I think is we can answer together is, do you expect that this would be useful for patients who are older, above the age of 20, and would it be stabilizing in terms of their respiratory or cardiac issues?
I strongly believe that to be the case. The first, and I have to tell you, it was astounding. This was a study run by a former president of the American Diabetes Association, who's a muscle expert. And he told me in his 40 years he has never seen the impact on diabetic muscle disease that he saw in that graph that I just showed you. Now, that's, again, small trial. But we have clear evidence that these were 75-year-old men, who are in end-stage heart failure, who were able to dramatically increase their muscle function. So we know that this is going to stabilize and improve function in older individuals. And I think part of the problem is that I mentioned to you that loss of mitochondria, which starts in one's mid-20s, is a universal feature of aging. Every 40-year-old man and woman has fewer mitochondria in their heart and skeletal muscle than they did when they were 20, which is about the peak age. So we have looked at this in everything from 20-year-olds to 80-year-olds and seen the same biology, which is evidence for improved muscle function and mitochondrial biogenesis. So we fully expect. I don't know how strong the effects will be, and I suspect that the longer the disease has progressed, the longer it's going to take to reverse. But I fully expect older populations to benefit from this.
Great, thank you. Thank you, George. And just as a reminder to those listening, the study that you did in the 75-year-old men with diabetes was done on the negative epicatechin. Is that correct, and not the positive epicatechin?
Yes, thank you. So we have nearly 80 patients in various disorders that have been tested worldwide with the minus form, which is the weakest form. We have, now this is also the only manufactured form of epicatechin that's been tested. So the trials with the plus, which is the material that you cannot find anywhere on the internet, and you cannot, it's very rare in nature. These are going to be the first clinical trials. Excuse me, I'm backing up. We have almost completed our first phase 1 trial with the plus at UC San Diego, and it's beautifully tolerated. We're seeing the same effect on follistatin and myostatin, and that study is about to finish. So we know it's safe, and so these Duchenne trials. And that was the one where the FDA approved our synthetic form of this. Our material is more pure than the average prescription drug on the market. And so that material is what we're now taking into the Duchenne population, and so we've had a safety trial supporting that, but this will be the first patients with illnesses that we put the plus version into. But because we're giving lower amounts of the plus because it's so much more potent, we're expecting, I mean, there really have been minimal numbers of side effects that we can identify, but we're giving actually less than we gave in the Becker's trial because the compound is so much more active. So we're anticipating fewer side effects with this next round of clinical testing.
Great, thank you. So, George, just a couple more quick questions as we come to the end of our hour. First, do you foresee any production problems with the plus Epicatechin? I think the last thing this community would need to hear without us getting your home address and finding you, as you might imagine, is we want to make sure that there is sufficient product available to do these studies as we go through them and hopefully as quickly as we are able to go through them.
Yes, that's a very good question. It's a very good question. We are manufacturing now at the 2 to 10 kilogram scale. That's roughly 20 pounds. This is very unusual for a biotech company because they can usually get enough for small trials, but they can't get enough for any broad treatment without partnering it. But my partner is a former head of medicinal chemistry or senior or vice president of medicinal chemistry at places like Johnson & Johnson. We have a FDA-approved manufacturing facility. We believe that we can easily manufacture this at the metric ton scale. So we are not anticipating any issues as we, other than the cost of making it, but we are not anticipating any issues as we go up into what would eventually be a worldwide treatment supply of drug. Unlike the biologicals for which this is, or the molecular biology drugs for which this is often a big problem, we're very, very comfortable that we can generate tons of this material as needed as our program expands.
Great. And George, the enrollment number for the trials that we're planning for the first trial and obviously broaden for the second, but start with the first, how many are we going to recruit?
We're going to be looking at 20 to 30, and depending on patient availability, it's just 2 months, and then we're going to be looking in the 50 to 60 range for the phase 2, 3 to 6 month efficacy trial. And so that and that will be placebo controlled, the phase 2 trial, with a crossover design, so that what we're hoping is that using the biomarkers in the first 2 months, if we see signs of activity like improved cardiac function on echo or on scan or improved muscle strength, that the placebos will then be crossed over to the active therapy group, so that ethically they'll be able, if there's evidence of clinical responsiveness, we're going to try to minimize their time on the placebo. And then we're going to basically drive right into a phase 3 trial where after we, of course, meet with the FDA and go for broke. I mean, just as I basically want this on the market in less than 4 years. So it's a question of how fast we see an effect, how durable we see an effect, and confirming that we don't see any potential for adverse events is going to make this thing go faster or longer. And obviously, with clinical research, those are the key questions. Right. But it's not going to be for lack of drug or lack of participating centers.
No, that's exactly right. The last question I'm going to answer is, will people who are non-ambulatory be able to walk? And I think what we know now about Duchenne is once you've lost ambulation, that involves not only muscles of the legs but muscles of the trunk as well. And it would be very difficult to restore muscle to that power, even with improving the mitochondria, to get someone out of the chair and walk. We wish for that. We're hopeful for that one day, but we're not The biomarkers that George is referring to is the work we've done with Larry Gold at SomaLogics out of Colorado. We have an ongoing study. The publications are with Synergy, Eric Hoffman, Yetrip Hathout's group, and this has been 4,000 circulating proteins that we measure by drawing blood, so there's no invasive in terms of biopsies. So we'll continue to do that work in collaboration with Yetrip Hathout and Lee Sweeney to make sure if we see a signal, we can change over from placebo. And so you will be hearing more. We are working on protocols. We're delighted to have this opportunity with you, George, and we really, as a community, are looking forward to moving as rapidly as we can. And our fingers crossed and are very hopeful that this, in fact, is one drug of part of a cocktail. PPMD is working on in the first quarter combination therapies. So you'll be hearing more about this. And again, hopefully this is one more tool in our arsenal. We're going to exploit it and explore it as completely and as rigorously as we can. You will be hearing more next year. And with that, I'm going to close and wish you all a happy holidays and thank you for everything this community does for us. Goodbye.